First of all, I would like to introduce myself. My name is Dr. Kundan Kumar Mishra. I am working as Assistant Professor, Department of Biotechnology at N.V. Patel Science College, Vallabh Vidyanagar, Anand. And here I will discuss, transcription, post-transcriptional processing, and reverse transcription for CSIR UGC NET preparation. In previous lecture, we had discussed on DNA damage and repair, but here we are going to discuss transcriptional, post-transcriptional processing and reverse transcription in details. First, we will discuss concept based on these particular topics and then some of the question which has been already asked in CSIR NET examination. So before I move for detailed discussion about transcription, first we have to understand the RNA molecules. There are two types of RNA. One is called coding RNA, and the example of coding RNA is messenger RNA, which are only present in 5 to 6% but highly diverse, and it is responsible for formation of all type of codons, and codons specify particular amino acid. Now second one is non-coding RNA, and that includes ribosomal RNA or rRNA, which has highest amount, approximately 80 to 85%, and it is one of the important structural components of ribosomes and protein. Another one is tRNA, which is also called transfer RNA or adapter RNA, and it is around 10 to 12%, responsible for feeding amino acid to the ribosome, means it carries anticodons, therefore it is called adapter molecules. Beside this, a lot of RNA is reported which are less than 1% in eukaryotes, particularly microRNA, a small nuclear RNA, SN, a small nuclear RNA, tmRNA, which is present in prokaryotes, then telomerase RNA and siRNA. These all RNA constitutes less than 1%, but responsible for processing as well as removal of introns during the process of post-transcriptional mechanism. Now the key concept of transcription is DNA is read from 5' to 3' to 5' during transcription. Complementary RNA is created from 5' to 3' direction. Only one of the two strands called the template strand is used for transcription because RNA is only single strand. Means duplex DNA strand, one will be called antisense, another will be sense. Sense is responsible for formation of functional messenger RNA, and that RNA will be single strand. The sense strand is sometimes called coding strand, which can specify codons. So far enzymes of transcription is concerned, then important enzyme is RNA polymerase. RNA polymerase is also called DNA dependent RNA polymerase. Reason is it requires DNA as a template, and it can bind it, and then it is responsible for formation of messenger RNA. Therefore it is called DNA dependent RNA polymerase. And these are the enzymes which never requires primer, means it can work without primer. There are different classes of RNA polymerase in pro and eukaryotes respectively. In prokaryotes, there is single RNA polymerase, which are responsible for formation of all type of RNA, whether it's rRNA or tRNA or mRNA or tmRNA. But so far eukaryotes is concerned, three different kinds of RNA polymerases are needed: polymerase 1, polymerase 2, polymerase 3. Polymerase 1 is responsible for expression of ribosomal RNA, all type of ribosomal RNA. Then second, polymerase 2, it is responsible for formation of messenger RNA, which specify codons, and polymerase 3, which are responsible for formation of transfer RNA. So in eukaryotes and prokaryotes, different type of RNA polymerases are there. In prokaryotes, single, and in eukaryotes, 3 type. That concept we will discuss later on, and it's a very important. Now what polymerase is required? Basically polymerase requires RNTPs instead of DNTPs. In DNA synthesis, there is a DNTPs, but in RNA synthesis, there is RNTPs. Then it requires cofactors like Mg2 plus, or if Mg2 plus is not there, then Mn2 plus can work. It requires DNA template as well as synthesis direction 5' to 3' direction. These are the requirement of RNA polymerases for transcription. So far mechanism of RNA polymerase is concerned, RNA polymerase proceed at much slower rate than DNA polymerases. As we have seen, DNA polymerase third added nucleotide 1000 nucleotide per second, while RNA polymerase can add nucleotides 50 to 100 bases per second. The fidelity of RNA polymerization is much lower than DNA, and RNA polymerase do not contain proofreading mechanism. That has to be remembered. If any mismatch are ribonucleotides added, then it cannot be corrected, and therefore it will leads to incorrect formation of incorrect protein, and sometimes this can create problem. We can see a structure of bacterial RNA polymerase. Bacterial RNA polymerase, there is a two parts. One is called core enzymes and another is called factors, which together form hollow enzymes. Core enzymes includes alpha, 2 molecules of alpha, 2 molecules of beta, beta prime and omega, plus sigma factor, and these together form hollow enzyme. Now this core enzyme is responsible for affinity of DNA, while sigma factor is responsible for decreasing the non-specific binding affinity of RNA polymerases. Now, so far eukaryotes is concerned, transcription is basically divided into three main parts: initiation, elongation and termination. Initiation includes recognition of RNA polymerase at specific promoters, and then some of the transcriptional factors, and then process of transcription start, that is called elongation. And when transcription complete, means when messenger RNA molecules form, then that transcription can be halted, called termination. So initiation, elongation and termination are the three main steps of either prokaryotes or eukaryotes transcription. But process of prokaryotes and eukaryotes is bit different. Differences is mainly lies on RNA polymerase second. It is more complicated multi-subunit enzyme. Eukaryotic genes are monocistronic, means single genes is responsible for formation of one particular polypeptides. The genes of eukaryotes are not continuous. There will be coding and non-coding sequence. If we will see genome of eukaryotes, approximately 98% sequences are considered as junk DNA or non-coding sequences, which do not specify any codons or any functional parts of the genes. That may contain introns, that may contain long terminal repeat sequence, that may contain transposon, retrotransposon, mini satellite sequences, micro satellite sequences, and only less than 2% are considered as coding sequences, which is responsible for specification of particular messenger RNA. So eukaryotes contain three different type of polymerases, as I discussed earlier, and each responsible for transcription for certain classes of genes. Here we can see this RNA polymerase 1, very important. Synthesize a pre-rRNA 45S, 35S in the case of yeast. This has to be remembered. This 35S has to be remembered, which matures into 28S, 18S and 5.8S. These are the components of ribosomes in the case of eukaryotes, 60S and 40S. And in the case of prokaryotes, it is 50S and 30S. RNA polymerase second, this synthesized precursor of messenger RNA and most small nuclear RNA and microRNA. RNA polymerase third, these synthesized transfer RNA, ribosomal RNA, and these 5S, very important, this 5S RNA and other small RNA. RNA polymerase fourth, which is not well characterized, but it is thought that it can synthesize a small interfering RNA, which has important role in gene silencing in the case of plants. And RNA polymerase 5, not well characterized, but synthesize RNA involved in siRNA directed heterochromatin formation in plants. So these type of RNA are only present in eukaryotes, not in pro. To understand complete transcription, we will have to understand the structure of promoter. Promoter are the sequence where RNA polymerase can binds and process of transcription start. And each RNA polymerases are highly specific for their own promoters. Therefore, each RNA polymerase recognize a specific RNA promoter for transcription, and there will be a structural variation between prokaryotic and eukaryotic promoter. Here we can see prokaryotic promoter at -35 region, which is a part of upstream regions. Upstream region is not part of functional form of genes. It facilitates the binding of RNA polymerase molecules at a specific site. So these -10 region is considered Tata box or Pribnow box, and these are the site where RNA polymerase binds. These are also called consensus sequences, and here RNA polymerase can binds, and these regions do not transcribe. +1 means downstream sequences are considered as transcriptional start site, and it has a structural genes. And then messenger RNA, here messenger RNA can start synthesis of codons. This one is prokaryotes. But so far eukaryotic promoter is concerned, the promoter is bigger one as compared to prokaryotes. It has cat box at -75 regions and Tata box at -25 regions. So these are the bit bigger, and here RNA polymerase, different type of RNA polymerase 1, 2, 3 binds, and process of transcription start from +1. These are the part of structural genes. Now, this DNA is duplex structure while RNA is single strand. So mainly RNA is single strand, and it is product of sense strands of coding sequences, and it is responsible for expression of all genes, means formation of proteins, and it has a structural and functional purpose or functional roles. While antisense RNA is specifically useful for variety of purpose in vitro condition, like if we want to prepare probe for hybridization to detect a specific RNA molecules on solid substrate like membrane, then it can be used like northern, or it can be used for location of particular transcript inside the cell through in situ hybridization, and it is one of the important part for the study of transcriptional events like nuclease protection assay. Beside this, when transcription completed, then reaction can be terminated, and transcriptional termination can be classified into two parts. One is called row dependent and another is called row independent. Two type of transcriptional termination reported. So far row dependent is concerned, it can be terminated by row factors, and these row factors move faster than RNA polymerase, and this can causes termination. Another one is row independent. Now these are the spatial region, these are the loop-like region in the DNA molecules which have GC reach and form a stem loop-like structure like transfer RNA, and causes messenger RNA detach from DNA template, and process of transcription halted. Now this one is very important. Many drugs designed based on transcriptional inhibitors, and the inhibitors are different. In the case of bacteria, rifampicin, it's a type of antibiotic which can block initiation of RNA chains by binding to RNA polymerase. This rifampicin is an antibiotic specific for inhibition of RNA polymerase in bacteria. These can inhibit bacterial RNA polymerase, means RNA polymerase, one type of RNA polymerase. In the case of bacteria, this can be inhibited, while some of the inhibitors which are specific to both pro and eukaryotes, like actinomycin D, these are effective for bacteria as well as eukaryotes. It can bind to DNA and block the movement of RNA polymerase, and process of transcription stop. And another important one which are only specific for eukaryotes and it doesn't have any effect on prokaryotes is alpha amanitin. Alpha amanitin is a chemical secondary metabolites which can extracted from Amanita phalloides, a fungus, which can block messenger RNA synthesis by binding to RNA polymerase second. It can preferentially bind to RNA polymerase second and stop the process of formation of messenger RNA. And if messenger RNA is not formed, then no questions of protein synthesis. So these three inhibitors are very important, and many of the questions have been asked in CSIR UGC NET so many times. Now come to another mechanism, what we call processing. As we know that in the case of prokaryotes, transcription occurs continuously, there will be no any processing events, and processing events is there, then that will be very slower. But so far eukaryotes is concerned, a lot of events occurs after transcription, and what we call this processing. Because of processing, we can get matured messenger RNA or transfer RNA or ribosomal RNA. When transcription occurs, then at pre-stage we can see on teletop, eukaryotic genes, if we will see, it has exons, introns and so many non-coding and coding sequences. Coding can be represented by C and non-coding can be N. So during the process of transcription, what happened? These coding sequences and non-coding sequences both transcribe, but non-coding sequences form loop-like structure, this loop, and this loop has to be removed by a process called splicing. So splicing is responsible for removal of introns as well as another non-coding sequences, and then exon molecules can be assembled together and mRNA will form. mRNA can form, but this mRNA is not ready for transcription. This can undergoes post-transcriptional modification. This can undergoes post-transcriptional modification where certain sequences has to be added, like cap at 5' end and polyetyl at 3' end. And then this messenger RNA, what we call post-transcriptional mechanism, and then this messenger RNA can transport into the cytoplasm for translation. So these events are basically absent in prokaryotes while present in prokaryotes. These RNA processing can be divided into 5 major types. Processing of nuclear pre-messenger RNA, as I had said. First, it will form hnRNA, what we call heterogeneous nuclear RNA. These hnRNA is heterogeneous nuclear RNA, and their introns has to be removed by a process called splicing. Loop has to be removed. There are different type of introns, group 1 introns, group 2 introns, and then if these will be removed, then functional messenger RNA will be formed, and after getting functional messenger RNA, processing of transfer RNA need modification of particular sequences. Highest modified bases are present in transfer RNA, while anticodon sequences are also there. So a stem and loop structure requires modification of bases for performing a specific function for protein synthesis. So processing occurs in tRNA, and processing also occurs in RNA for folding in correct configuration for protein synthesis, and these all events occurs in nucleus, and then messenger RNA can be transported into cytoplasm for protein synthesis. One more mechanism important which has changed the concept of central dogma in molecular biology is reverse transcription. It was thought that flow of genetic information is unidirectional, means genetic information is passes from DNA to protein. But it was Temin and David Baltimore in 1964 found that in certain retrovirus there are presence of specific enzymes, what we call reverse transcriptase, which has ability to use messenger RNA as a template and convert into cDNA by reverse transcription, and that opens the door of new chapter in the area of central dogma, what we call central dogma reverse. Although this retrovirus is specifically present in many retrovirus, reverse transcriptase enzyme, but it has ability to convert any messenger RNA molecules into cDNA, whether messenger RNA is isolated from human beings or whether messenger RNA is isolated from plants or in different condition, either a stress condition or non-stress conditions. So these are the important point to locate the presence of coding sequences in our genome through reverse transcription. We can isolate messenger RNA from different cell for different period, for different conditions, and then we can convert into cDNA. So whatever coding sequences or gene expressed on that time, that can be analyzed. So this was the one of the major breakthrough in the area of differential gene expression studies or functional gene expression studies in eukaryotes, particularly because eukaryotes has 98% sequences which do not specify any codons or consider as non-coding sequences or junk DNA. Only 2% or less than 2% sequences had functional role. So in eukaryotes, it was one of the major breakthrough, and for that, Tevin and David Baltimore were awarded Nobel Prize for this discovery. Reverse transcription is also called RNA-dependent DNA polymerase. It has three activities. Mainly it possesses three activities. First, it can utilize messenger RNA as a template and convert into cDNA. Then this cDNA can be converted into duplex DNA. And before conversion of duplex DNA, messenger RNA molecules from DNA-RNA hybrid is also degraded by this reverse transcriptase. So it has three activities. It has three main functions. First one is reverse transcription. Then it acts as a nuclease. It can remove RNA from DNA hybrids, and then it can act as a polymerase, means it can utilize single strand cDNA as a template and synthesize duplex DNA molecules, and therefore it is one of the important enzyme in the area of molecular biology and biotechnology. Here we can see a retrovirus which has a specific RNA molecules as a genomes, and this attached to the specific site of a particular cell and release its RNA molecules, and reverse transcriptase enzyme can convert this viral RNA into DNA, and this can integrate into our genome and it will behave as a part of our genome. The majority of retrovirus is responsible for cancer in human being, like human T-cell leukemia virus, human T-cell lymphoma virus. These are the present in our genome and responsible for cancer because it has certain genes, and that genes when integrated at a specific location in our genome, then new combination of genes will be there. And if it can be integrated at proto-oncogenes, then that proto-oncogenes can be activated into oncogenes and it is responsible for causing cancer. But this retrovirus is beneficial if we will remove the oncogenes from their genome. Then this can be used as a vector, and this can be used as a source material for transferring or transfecting a specific genes into mammalian cell, because usually retrovirus do not kill their host. These things I had already discussed. It is called RNA-dependent DNA polymerases, and RNA-dependent DNA polymerase, RNA hydrolyze, means it can hydrolyze RNA molecules from cDNA RNA hybrids, and it is also called DNA-dependent DNA synthetase or DNA polymerase. Now come to the important aspects, and that is questions from transcription processing. Now here first question is, during transcription, if the nucleotide sequence of DNA strand that is being coded is ATACG, then the nucleotide sequence in the messenger RNA would be, here we can see on teletop, the sequence is ATACG. This is sequence of DNA. So obviously if we want to sequence mRNA, then we know that in the case of DNA, A pair with T while G pair with C. But so far mRNA is concerned, mRNA is single strand, but transcription occurs in this way, कि instead of T there will be U, and instead of A there will be U. So what happened? Instead of A here will be U, and instead of T, here will be A. So A again U, C here, G and G C. This one is okay. So instead of T there will be U, and instead of A there will be U. So we can find the sequence. This A U T A, thymine is replaced by adenine, adenine is replaced by uracil, cytosine is replaced by guanine, and guanine is replaced by cytosine. So this C is correct. T is not present in RNA at all. So both A and B option is completely wrong. And D, here we can see T. So T is not part of RNA. T is not present in RNA. So answer is C. This one is correct answer. Next one. Which form of RNA has a structure resembling clover leaf? You know, so far a structure of ribosomal RNA is concerned, it's a highly folded structure. Very difficult to predict because of highly folding. Heterogeneous nuclear RNA, this is considered as a part of messenger RNA in prematured condition. When transcription occurs, then it has lot of other sequences which can transcribe and which further removed by splicing. So it is a mixture of exons as well as some of the another sequences, and that sequences has to be removed. So heterogeneous RNA has linear structure. It's a linear structure. Messenger RNA, this mRNA has linear structure. In eukaryotes, this messenger RNA has cap at 5' end and poly A tail at 3' end, and it is monocistronic by nature. So this is linear one, and it carries codons. But if you will look tRNA, then tRNA has secondary as well as tertiary structure. It can folded in many configuration, and it can folded into a special type of a structure, what we call a stem and loop like a structure. Here it can appears like this. So this can appears like clover leaf. Clover leaf, and here anticodon region is there, and at 3' there will be CCA sequences present which can carries amino acid. So this transfer RNA is basically has clover leaf structure. Next one, very important one. RNA polymerase second from eukaryotes is inhibited by. We know that RNA polymerase is responsible for formation of messenger RNA molecules, and therefore it has very important role. And many drugs for treating particular virus and even HIV is based on inhibitors of RNA polymerases as well as reverse transcriptase. One of them is alpha amanitin. This alpha amanitin is extracted from a fungus what we call Amanita phalloides. Amanita phalloides is a special type of fungus which has toxin, and that toxin is called alpha amanitin, and it has a special affinity with RNA polymerase second. If we will see the structure of RNA polymerase, it has a special affinity. It can bind at the active site and remove the process of transcription. So here this one is correct, alpha amanitin. Rifampicin is antibiotic which can inhibit RNA polymerase, bacterial RNA polymerase, and bacterial RNA polymerase is single type. Another one is streptomycin. It's antibiotic. It doesn't have any correlation with inhibition of transcription. It can inhibit translation. And another one is cyclohexamide. It can inhibit replication. It doesn't have any correlation with transcription. So alpha amanitin is right answer, and this question has been asked in NET many times. Now next question is basically from post-transcriptional modification. Capping. What is capping? Capping of messenger RNA. Why capping present in mRNA, and what is capping? Actually, if we will see messenger RNA of eukaryotes, it has very short lifespan. Plus, it is highly sensitive against RNA age enzymes. It can degrade RNA very rapidly. Therefore, cell has developed certain technique to protect this mRNA against RNA age, and for that two type of events occur. One is called polyannulation, where poly A polymerase added poly A tail at 3' end, and another one is addition of guanine at 5' end to protect messenger RNA against RNA age enzymes. So capping of messenger RNA means addition of guanine residue at 5 prime of messenger RNA molecules so that it's get protected against RNA age and specify whatever functions. So first option is RNA polymerase 1 generated transcript. It's wrong. Transfer of phosphate from 3' end of nascent RNA. This can be done by alkaline phosphatase, and alkaline phosphatase cannot work on RNA. So this option is wrong. HNRNPs, heterogeneous nuclear RNAPs, is wrong. And addition of 7-methylguanosine to the 5' end is right. Where RNA becomes stable and it can protect it against RNA age. Next question has been asked in net so many times. There are 3 kinds of RNA polymerase 1, 2, 3 in eukaryotic cell, and each is specific for one class of RNA molecules. Which of the following is correct match? As I said you earlier, if you remembered what I said, there are 3 different type of RNA polymerase in the case of eukaryotes. One is responsible for formation of ribosomal RNA, 2 is responsible for formation of messenger RNA, and 3 is responsible for transfer RNA. So if we remember this, then we can see the option. First one, there are 3 kinds of RNA polymerases in eukaryotic cell, each is specific for one class of RNA molecules. Then which of the following is correct match? RNA polymerase 1, rRNA true. RNA polymerase 2, tRNA is wrong. Here third is required. RNA polymerase 2, mRNA is true. RNA polymerase 3, RNA is wrong. Here is tRNA. RNA polymerase 1, rRNA is right. RNA polymerase 2 is messenger RNA is right. RNA polymerase 1 tRNA is wrong. RNA polymerase 3 is rRNA is wrong. So answer is C. RNA polymerase 1 rRNA and RNA polymerase 2 mRNA. Next one is concept based and very good question. Question is, a retrovirus is an RNA virus with the ability to convert its sequence into DNA by reverse transcriptase. Reverse transcription is mediated by enzyme reverse transcriptase. Which of the following are the function of reverse transcriptase in retrovirus? I had already discussed, and question is based on that fact. First one is, it hydrolyzes the host cell's DNA. Not true. Retrovirus, whenever retrovirus, we can see on teletop, suppose this one is host and this one is retrovirus having RNA as a genome. When binds at the surface, first it's released its RNA genomes, and this RNA genomes is converted into cDNA by reverse transcription. So it never hydrolyzed host cell's DNA. Host cell's DNA is never hydrolyzed. It uses viral RNA as a template for cDNA synthesis. That's true. This one is viral RNA and this can be converted into cDNA by reverse transcription. So second point is right. Third one is convert host cell RNA into viral DNA. No, host cell DNA is already present in nucleus and there is no any correlation of degradation of host cell DNA. It translates viral RNA into proteins. It translates viral RNA into proteins. No, reverse transcriptase doesn't have that function. It never translates viral RNA into protein. Viral RNA is translated by host cell protein synthesis machinery. Then it uses viral RNA as a template for making complementary RNA strand. Never. RNA converted into cDNA. So there is no questions of RNA template, not at all. Then it degrades RNA from DNA RNA hybrid. That's true. During the synthesis of first what happened, suppose this is RNA and this RNA first converted into cDNA. So what happened here? mRNA cDNA hybrid is there. This mRNA cDNA hybrid is there. So this RNA can be degraded by reverse transcriptase. this RNA can be degraded, and that is called degradation of RNA from DNA RNA hybrid. So these two, it uses viral RNA as a template for DNA synthesis, and it degrades RNA from DNA RNA hybrid is true. So option is 2 and 6 rather than all these 4, 5. These two are the facts, has to be remembered. These 2 and 6. Next one is very important, ribozyme. This was discovered by Tom Keck, and this is one of the concept of processing of RNA molecules. RNA act as an enzyme. One question always in our minds, which come first, whether DNA or RNA, and there are plenty of evidences indicate that RNA first come and then DNA evolve. Like RNA has ability to transform into cDNA, as we have seen in the case of reverse transcription. Another one is RNA act as an enzyme. During the process of splicing, RNA itself responsible for trans-esterification reaction, where non-coding sequences is removed by degradation, and that is done by RNA itself. That means splicing is performed by RNA itself. There is no any another supports. There is no another enzymes. Although RNA is responsible for formation of proteins, and that proteins act as a functional enzyme. That is different issue. But here RNA itself plays important role as an enzyme. Therefore, scientists said that RNA possesses 3-4 features. It a genetic material in certain retrovirus or RNA virus. It has ability to convert RNA into cDNA, means it can transform into DNA. It acts as an enzyme. Therefore, RNA evolved first. And if we see that concept, then this question is very important. Ribozyme is, what is ribozyme? RNA itself acts as an enzyme called ribozyme, and sometime we called it catalytic RNA. So ribozyme is catalytic RNA which catalyzed both synthesis and degradation of a small oligonucleotides during the process of splicing. It's not derivatives of vitamin riboflavin. It's not true. Name of the scientist who discovered ribosome. It's not true. It's an antisense strand of DNA. It's not true. Ribozyme is catalytic RNA. Next question is concept based, very important. When a growing replication fork encounters an RNA polymerase complex engage in transcription. See the question carefully. When a growing replication fork, we had already seen replication fork previously, an RNA polymerase complex engage in transcription, means replication fork is moving. Along with that, RNA polymerase synthesizing RNA molecules. Then what happened? We can see the option. Both DNA replication and transcription stop. No. Replication can be only terminated when there will be formation of TUS-TER complex, or when there will be a specific sequences where terminus utilization substances bind and replication fork halted. So this one is not true. Replication stop but transcription continue. Again, this is not true. Replication only stop when TUS-TER complex form at a specific sequence, and there is no specification of that sequences. Then next option is both DNA replication and transcription continue but slowly. This is not true. Replication is not bypassed by transcription. This can occur one after another. So what happened? Next one, transcription pause to allow replication work to progress. This is true. Actually, we know that first DNA has to be replicated. If DNA is not replicated, it will not transcribe, it will not post-transcriptionally active, and it will be not synthesize a specific protein again required for DNA synthesis. So obviously, what happened? Transcription will pause, process of transcription will stop, and then replication will first proceed, it will complete, and then it will allow transcription to occur. So last option is correct one. D, transcription pause to allow replication fork to progress. Next one. Again, this one is good questions. Among the following, which statement is correct regarding the promoter of gene in eukaryotes? This question is from eukaryotes, and we have seen the differences between prokaryotic transcription and eukaryotic transcription. Among the following, which statement is correct regarding the promoter of gene in eukaryotes? First one, it is always located upstream of transcription at start site is not true. We had already discussed on promoter sequences. Then it is always located downstream of transcription at start site. Again, it's not true, not always. Different promoter elements are recognized, different RNA polymerases. We should remember what I said. RNA polymerases are highly specific to their own promoter. If promoter sequences will be changed, then transcription will not possible. Like, suppose I have a gene, a specific genes, and if their promoter will be not there, and if we want to transcribe it in vitro condition, it will not transcribe because RNA polymerases are highly specific to their own promoter. Like, if we want to express a particular gene in vitro condition, like human genes, if we want to express in vitro condition, it must be fused with a specific promoter, and then we can select RNA polymerase accordingly. Like T4 or T7. These two are the bacteriophage. If we will isolate their promoter and we will fuse the genes of human being with T7 and T4 and add T7 or T4 RNA polymerase, then that gene will be transcribed. Means what it indicates? Ki each RNA polymerase are specific for a specific promoter, and promoter sequences are highly specific. Therefore, these options 3, different promoter elements are recognized by different RNA polymerase. This one is true. Promoter are recognized due to their different secondary structure is again wrong. C option is perfectly right. Next question. In bacterial RNA synthesis, which of the following is function of factor Rho? I had already discussed. There are two parts of enzymes in the case of RNA polymerase. One is called core enzyme and another is called hollow enzyme. These allow proper initiation of transcription. These eliminating the binding of RNA polymerase to the promoter, participate in the proper termination of transcription, and increase the rate of RNA synthesis. Allow proper initiation of transcription is not true. Eliminating the binding of RNA polymerase to the promoter is again not true. It can participate in the proper termination of transcription is right. RNA synthesis, row dependent and row independent terminations are there. Transcription can be terminated in two different ways. One is row dependent and another is row independent. In row independent, a specific stem and loop like structure form, and that has G series sequence which can halted RNA polymerase. So row factor has participate in the proper termination of transcription rather than initiation, as well as increase the synthesis of RNA molecules. This question is again concept based. If you were designing a method to specifically inhibit prokaryotic transcription but not eukaryotic transcription, interfering with which of the following would work best? This question is very important. If you were designing a method to specifically inhibit prokaryotic transcription but not eukaryotes, means our aim is to inhibit prokaryotic transcription and allow eukaryotic transcription to occur, then we will have to select, or we will have to put our apply our brain ki which type of events can be altered so that prokaryotic transcription can stop but eukaryotic transcription can continue. One thing is inhibitors. As I discussed, some of the inhibitors are specific to prokaryotes and some are eukaryotes and some are both, like rifampicin. it's a specific for prokaryotes. Alpha amenity, it's a specific for eukaryotes. And few others are like actinomycin D. It is a specific for both bacteria as well as eukaryotes, means pro and eukaryotes. So here options is not that. Option is DNA ase activity. If we will add DNA ase enzyme, it can degrade all DNA molecules. So automatically both prokaryotic and eukaryotic transcription will not possible. Then recognition of promoter region. Yeah, this one is true. This one is true. Because promoter are highly specific to their genes. And prokaryotic and eukaryotic promoter, I had already discussed, their structure is different. And their RNA polymerases is different. So if we will devise two different type of promoters, then what happen? If we will add a specific RNA polymerase, then that transcription will occur, other will stop. Like if we will take eukaryotic promoter and add eukaryotic RNA polymerase, then automatically prokaryotic transcription will not possible. Third one, an intercalating agents, it can intercalate between the DNA molecules, and both prokaryotic and eukaryotic transcription will not possible. And ribosomal binding is protein synthesis machinery, it will not work. So answer is recognition of promoter regions. This one is memory based, not concept based. 3 of the 4 eukaryotic rRNA are synthesized from single transcription unit consisting of rDNA. Which of the following does not belongs to this group? Eukaryotic RNA. If we will classify eukaryotic RNA, actually eukaryotic RNA can be classified into 2 main groups on the basis of Sedberg unit, 60S and 40S. And each 60S and 40S is composed of a specific ribosomal RNA. Like if we will talk about 60S, there is 28S, 5.8S. And if we will talk about 40S, there is 18S. 5S is component of a small subunit of prokaryotic rRNA. So what question says? 3 of the 4 eukaryotic RNA are synthesized from single transcription unit consisting of rDNA. Which of the following does not belongs to this group? Obviously 5.8S, this belongs. 18S, this belongs. 28S, this belongs. So which one is left? 5S. So answer will be B, 5S. Now again, here is concept based questions. And which of the following events does not occur during pre-transfer RNA processing? We remember RNA processing. And what happened in RNA processing? First, messenger RNA has to be processed. And processing of messenger RNA, 2 events is there. One is addition of poly-A tail at 3' end, what we call polyannihilation, and another is addition of modified guanine residue at 5' end, what we call capping. These 2 are only occurs for messenger RNA, not for ribosomal RNA and transfer RNA. Now here we can see option is 5' end cleavage by the RNA ASP is true. Addition of poly-A site. This poly-A site is not added in tRNA. Poly-A tail site is only added in mRNA. Addition of CCA sequence at the 3' ends is perfectly right. If CCA is not added, then it cannot be transport amino acid sequences for protein synthesis. And transfer RNA has highest chemically modified bases like pseudouridine. These are the present, 60 or 70% bases are modified in the case of tRNA for performing protein synthesis. So if we will see, then this option is right. Addition of poly-A tail, poly-A site, is right. It is not a part of tRNA processing. Now next question is again important, and here is more than one option. Which of the following is true about poly-A tail? I had repeated so many times, poly-A tail, poly-A tail. We will have to select. It is synthesized post-transcriptionally, is true. Perfectly right. Then, it is usually shorter than 300 nucleotides. Actually, its range is from, what happened? We can see here. When this messenger RNA is synthesized from DNA, then polyetyl is added by a poly-A polymerase, and it has a specific processivity and turnover number. So after 200 to 300 base pair, it automatically its turnover number stops and processivity gone. So length of sequence will be 300 nucleotide, is again true. One of its function is to protect the messenger RNA from cytoplasmic RNAs, again true. One of its function is to stimulate translation, very true. So polyetyl has all four functions. Therefore option is P, Q, R and S. All fours are right. All four options are right. So answer is D. Oh, this one is big one, but we had already discussed the facts. Rifampicin, it's antibiotic, is a bactericidal antibiotic drugs used typically in treating mycobacterium infection. Mycobacterium tuberculosis is responsible for tuberculosis in human being and in other animals. Which of the following statement describe the mechanism of action of rifampicin? I had already said to you that this rifampicin is responsible for what? Inhibition of RNA polymerase in bacterial cell. But what way? First one is rifampicin inhibit DNA dependent DNA polymerase in bacterial cell, DNA dependent RNA polymerase in bacterial cell by binding its beta subunit, thus preventing transcription to RNA. Is very right, perfectly right. Interfering with translation through binding to the 30S ribosomal subunit is wrong. It doesn't have any relationship with protein synthesis. Interfere with the cell wall biosynthesis of growing bacteria, not at all. Only one antibiotic, what we call ampicillin. Ampicillin kills bacteria by destroying cell wall. So this rifampicin is not has that action. And interfere with 70S ribosome, resulting in incorrect translation of mRNA. Again, it doesn't have any relationship with protein synthesis. So option 1, rifampicin inhibit DNA dependent RNA polymerase, that is RNA polymerase in bacterial cell by binding its subunit, thus preventing transcription to RNA, is right. This one is again simple question. Which of the following is characteristic of eukaryotic transcription but not prokaryotic transcription? Means we will have to select which events is exclusively occurs in eukaryotes, not prokaryotes. Transcription and translation are coupled. Transcription and translation are coupled. Which of the following is characteristic of eukaryotic, while this is characteristics of prokaryotes? Getting it? There is one form of RNA polymerase. This is characteristic of prokaryotes. RNA polymerase second is responsible for synthesis of messenger RNA. Very true. RNA polymerase 1, rRNA, polymerase 2, mRNA, polymerase 3, tRNA. So this option C, RNA polymerase second is responsible for synthesis of messenger RNA, is 100% right. While RNA polymerase is polycistronic, is not true. RNA can be polycistronic, is not true. RNA is monocistronic in the case of eukaryote. So option is C. Option is C. RNA polymerase second is responsible for synthesis of messenger RNA. Now next one. It's a processing or post transcriptional modification. Which of the following is not true of RNA processing? Means we will have to select the correct options. First one, introns are excised out before messenger RNA moves out of nucleus. I had already spoken to you many times. I had already discussed that first introns has to be removed at a specific site through splicing, and for splicing RNA itself acts as an enzyme. The stem and linear and loop like structure form, and that structure is called heterogeneous nuclear RNA. So loop has to be removed first, and then exons will be assembled together and functional messenger RNA form in the case of eukaryotes, and then it can be post-transcriptionally modified through capping or polyadenylation or etc. So this one, introns are excised out before mRNA moves out of nucleus, is true. Sorry, introns are excised out before messenger RNA moves out of nucleus is not true. Introns are excised before mRNA moves out of nucleus is not true. Very right. The presence of introns may facilitate crossing over. There is no correlation of crossing over between these processing. So wrong. RNA splicing may be catalyzed by a spliceosome. RNA splicing may be catalyzed by a spliceosome. No, spliceosomes are group of factors, but RNA splicing is catalyzed by RNA itself, what we call ribosome. So this is not true. A primary transcript, this is true. A primary transcript is often much longer than the final RNA molecules that leaves the nucleus, is true. So first one, introns are excised out before mRNA moves out of nucleus is not true so far processing of RNA is concerned. Rest one is true with certain cause and limitation. One more questions is straightforward. A post-transcriptional processing events that occurs in the formation of both messenger RNA and transfer RNA in eukaryotes is, we have to consider both RNA, tRNA and mRNA together. rRNA is not there. So we will have to consider a structure as well as processing of both RNA in mind, and then we will have to select proper option. A post-transcriptional processing events that occurs in the formation of both messenger RNA and transfer RNA in eukaryotes is the addition of sequence CCA to the 3 ends. This is characteristic features of tRNA. mRNA is not there. Then the removal of introns by splicing. This is features of messenger RNA, not transfer RNA. The modification of some of their bases. This is shared by both tRNA and mRNA. If we will see the events of processing, mRNA needs poly-A-tail and capping at 5' and 3' and respectively, what we call modification of bases. tRNA requires addition of CCA at 3' end for transfer of amino acid to the growing chain of polypeptide, as well as it has another pseudouridine or some of the bases to carries anticodon and codon interaction. So modification is both shared by tRNA and mRNA. So answer is C. C1 is right answer. The modification of some of their bases. Cleavage events before polyadenylation is not true. C is the right option, and this question is asked twice in NET exams. This one is very simple one and very straightforward question. Eukaryotic messenger RNA differ from prokaryotic messenger RNA in what way? Eukaryotic messenger RNA differ from prokaryotic messenger RNA in what way? I had already discussed. Eukaryotic messenger RNA is basically monocistronic, while prokaryotic messenger RNA is polycistronic. Beside this, some of the modification is on their promoter sequences. So we have to keep those points in our minds. First one, they do not have 5' untranslated region. This is not true. Their coding regions are separated by spacer. Yes, we can see. Eukaryotic messenger RNA constitutes heterogeneous nuclear RNA in the form of some of the spacer like introns or some of the junk DNA, while prokaryotes do not. Third option, they do not have 3' upstream translated region. This is not true. They have a free 3' hydroxyl group on their end. This is not true. So this one is right. Eukaryotic messenger RNA differ from prokaryotic messenger RNA that their coding regions are separated by spacer. Coding regions are separated by spacer means intron sequences and some of the junk DNA sequences. Next question. It is again based on concept. As I said if we will see polymerase, any polymerase, whether it's DNA polymerase 1, 2, 3, 4, 5, reverse transcriptase, Taq DNA polymerase, all polymerases required primer. Only one polymerases, which are exception, which can synthesize nucleotides in 5' to 3' direction and never requires primer, is RNA polymerase. And therefore, you will find some of the question is NET and other examination, they are repeatedly asking this type of questions, and we can see this question: which enzyme does not require primer? RNA dependent DNA polymerase, means it will be depends on RNA and synthesized DNA. It's a features of reverse transcriptase, is not true. DNA dependent DNA polymerase, of course template will be DNA and product will be DNA, so DNA polymerase, it required primer, so it's not true. DNA dependent RNA polymerase means template will be DNA and product will be RNA, so this one is perfectly right. C, DNA dependent RNA polymerase doesn't require primers, while Taq DNA polymerase, it's a type of DNA polymerase, it requires primer. Transfer RNA genes are transcribed by, this one is very straightforward questions. There is no need of any concept. 3 type of RNA in eukaryotes I had already discussed, 1, 2, 3, and each will be specific for expression of particular RNA, and their structure is bit different. Okay, 1 rRNA, 2 mRNA, 3 tRNA, and here if tRNA is there, then transfer RNA genes is transcribed by polymerase 3rd. Reverse transcriptase, there is no correlation, it is responsible for reverse transcription. Next one, reverse transcriptase is again a straightforward question. It's RNA dependent DNA polymerase and DNA dependent DNA polymerase. RNA dependent RNA polymerase and RNA dependent DNA dependent RNA polymerase. Now here what I said you earlier, this reverse transcriptase using RNA as a template and convert into cDNA, and there will be formation of hybrid like structure, means mRNA and DNA both sequences will be there. So mRNA sequence has to be removed to get duplex DNA. Without duplex DNA, it is not possible to transcribe them, because subset of transcription is duplex DNA rather than single strand. So even if we find coding sequences of any genes, we require duplex DNA, and for that there should be degradation of mRNA molecules from cDNA. So reverse transcriptase using RNA as a template, it can degrade RNA and it can synthesize duplex DNA from single strand. So here if we will see the options, it's RNA dependent RNA polymerase and DNA dependent RNA polymerase is not true. Again, RNA dependent DNA polymerase and DNA dependent RNA polymerase is not true. RNA dependent DNA polymerase and RNA dependent polymerase is not true. So option is RNA dependent DNA polymerase, means template will be RNA, it will form cDNA and then it will synthesize complete DNA, duplex strand. So A is right answer, RNA dependent DNA polymerase and DNA dependent DNA polymerase. One question is from termination. How replication terminated? Although we had already seen row dependent and row independent termination, and there are row factors involved in row dependent and row stem loop structure involved in row independent, and what we call attenuation or anti-terminations. The concept of attenuation and anti-termination is based on that. In E. coli, means bacteria, attenuation and anti-termination utilize which structure? We will have to find the structure. And what I said earlier, the row independent termination form loop and stem like a structure, and if we will remove this stem and loop like a structure, then process of transcription will be continue. Okay, in row independent things is different, but in row independent only G series sequence form a structure. So a stem loop structure in RNA is true, a stem loop structure in DNA, no, it's wrong. RNA DNA hybrids in the case of cDNA formation and differential protein folding, it doesn't have any correlation with anti-termination. So answer is a stem loop structure in RNA. Here next question is based on concept, and more than one option is required. Therefore, we will have to see all the options. Which of the following statements are correct about spliceosomes? A spliceosome is needed for splicing of RNA molecules, and lot of U-rich sequences are there. Lot of factors will be there, and RNA start removing introns. And a spliceosome is responsible for removing of both group 1 and group 2 introns. So we will have to select which one is right. Which of the following statement or which of the statement are correct about a spliceosome? P, are composed of RNA and proteins, is very right. Spliceosomes are basically composed of both RNA as well as group of proteins. Recognize RNA sequences that signals for removal of introns, is very true. This is function of spliceosomes. It can produce different messenger RNA molecules by splicing and alternate site, is again true. Non-coding and coding sequences or junk DNA can be differentiated, and mediates the splicing of pre-transfer RNA, is again true. So whatever option depicted, P, Q, R, S, all are true, and therefore correct answer is D, P, Q and R. Next one, it's very simple questions. Very simple, anyone can do it. If you have even a little bit concept of transcription, Tata box and Privno box are components of. In the case of prokaryotes, -10 and -35 regions, I had already discussed what is called upstream sequences are basically Tata box or Privno box. So it is component of what? Operators, not true. Promoters, true. Enhancer, not true. Activator, not true. So Tata box or Privno boxes are components of what? Promoter. Now this is all about of transcription and post-transcriptional mechanism. I had tried my level best to give the sufficient reasons why this option is correct and why another option is not correct. Beside this, I had covered some of the fundamental concept within defined period of time. I hope this lecture will be fruitful for all NET aspirants, and I wish you all the best. Thank you very much.